Right, so we'll continue with the topic of control of gene expression in eukaryotes. We'll be looking at control at the transcriptional level in this video. Okay, so for the gene that is found in euchromatin, which we have looked at in the previous video, this gene is able to be transcribed. But that doesn't mean that transcription will always occur, right? Transcription will occur only when needed. So at the transcriptional level for a gene found in the euchromatin, it controls exactly when is this gene transcribed and how much transcription will occur for this gene. Okay, so for the transcription to occur, right, for a particular gene, as mentioned earlier, the gene must be found in euchromatin. Okay, and also there must be the binding of certain transcription factors and also the RNA polymerase to the specific sites on the DNA to initiate transcription. For transcription factors, right, there are two kinds or two categories. Okay, you have the general transcription factors, which you have learned before under transcription in protein synthesis. And there are another category called the specific transcription factors. And under this category, there are two types. We have the activator and the repressor proteins. Okay. As for the specific sites, right, you have learned about the promoter sequence before. So in this topic now, we will be looking at two more sequences called the enhancer and the silencer sequence. All right, so these sequences together are known as the control elements. I think I've typoed there, yeah? Control elements, missing an E. So anyway, right? The activator protein, well, will bind to the enhancer sequence. The repressor protein will bind to the silencer sequence. The general transcription factors, as you have learned before, they bind to the promoter sequence, recognizing the Tata box, right, in the promoter sequence. All right, so let's do a bit of differentiation between the different control elements. All right, so as mentioned before, I've already talked about which transcription factor binds to which control elements, right? But as for the location of the control elements, the promoter sequence is just before the gene that it controls. So it is very close, in other words, proximal. It is proximal to the gene that it controls. It is before the gene, right? So upstream of the gene. As for the enhancer and silencer sequences, right, they are a bit different. They are very far away from the gene that they control, right? So that's the distal, very far away from, at least relatively speaking, right, compared to the promoter sequence. And they don't have to be before, just unlike the promoter, they don't have to be before the gene, right? They can be before, can be after, because they're so far away, right? So whether it is before or after, it doesn't really matter. All right, so it can be upstream or downstream of the gene that it controls. The enhancer and silencer sequence may not even be on the same chromosome as the gene that they control. Okay, but in the lecture, right, I'll be looking at the enhancer or silencer sequences that are on the same chromosome. Now that makes it a bit simpler. Okay, but at the back of your mind, you would need to, or you should know that the enhancer or silencer sequence may not even be on the same chromosome, right? Okay, so can be from another chromosome, but then the enhancer or silencer sequence is like brought close to the promoter sequence for some interaction. Later you will we look at the interaction a bit later, right? So anyway, let's look at how the control elements work together, yeah, to regulate transcription. Okay, so as mentioned, we have the promoter sequence, right, which is close to the gene, right, so just before, just upstream of the gene. And then we have the enhancer or the silencer sequence, right? So first of all, just to clarify, right, a particular DNA sequence cannot have two functions. So a particular DNA sequence may be the enhancer, or it may be the silencer. Okay, it cannot be both. Yeah, so just to clarify that, so don't be confused by the diagram. Yeah, so this is either an enhancer or a silencer. Oh. Okay, so anyway, you see all this dash, right, slash, right? It means the enhancer or silencer sequence is very far away from the gene here. Okay, so what happened is the general transcription factors would bind to the promoter via the Tata box and would then recruit the RNA polymerase to form the transcription initiation complex that you have learned before under protein synthesis. As for the enhancer or silencer sequence, right, as mentioned just now, activator proteins would bind to the enhancer and repressor proteins would bind to the silencer. Right? So if this is a enhancer, then an activator protein would bind. If this is a silencer, then a repressor protein would bind. Okay? Binding of the activator or repressor, right, would cause the DNA to bend or loop with the aid of other proteins called DNA bending proteins, as you can see here. Okay, and that would bring the activator or the repressor close to the promoter, right, close to the transcription initiation complex, and then the activator or repressor would be able to interact with the transcription initiation complex. This interaction would either increase the rate of transcription, if not decrease the rate of transcription, depending on whether it is an activator or a repressor. Alright, of course, the repressor would have other mechanisms to downregulate transcription as well, which we'll look at slightly later. Alright, but this is one mechanism. Okay, so as I mentioned just now in the previous slide, right, this diagram shows you a enhancer or a silencer that's on the same chromosome, right? This is the same DNA molecule, right? So the enhancer or silencer can also be on a different chromosome. So instead of this DNA bending, what happened would be that somehow the enhancer or the silencer, right, from another chromosome would be brought close to this particular promoter here to interact with this particular transcription initiation complex. Right, so bringing the activator or the repressor bound to the enhancer or silencer close to the transcription initiation complex still happen, just that it is happening with a different mechanism instead of just a normal DNA bending, which is shown here, right? That it will be on the same chromosome. Okay, so yeah, just a little bit of extra stuff there. Okay, so anyway, right, there will be interaction between the proteins, which would affect the function of the transcription initiation complex. Okay, right, so to combine with our first video, right, in terms of chromatin level control, right, in order for us to talk about the binding of all these transcription factors and RNA polymerase to the DNA, your DNA must be loosely packed. If not, there's no binding, right? As we have talked about before, right? There will be like no access of the proteins to the DNA. Okay, so the DNA would be in the form of euchromatin, and that allows the enhancers and or the repressors to be bound by the activator or repressor proteins that allow the promoter to be bound by the general transcription factors and RNA polymerase. Okay, so then, right, depending on which protein binds to the sites, right, whether it is the activator binding to enhancer or the repressor binding to the silencer, right, that would affect the rate of transcription. Okay, so both the chromatin structure, right, the control of the coiling of the chromatin, as well as this transcription control by the activator or repressors, both controls transcription. So you might be thinking, like, what's the difference, right? The difference is the control of the chromatin coiling, right, to control transcription. This form of control is more permanent, right, more long-term. Because if you think about, say, the pancreas, right, it's an example that I like very much. Right, the beta cells makes insulin, so the beta cells express the insulin gene, but not the glucagon gene, right? They don't make glucagon, right? So for the beta cells, because they would never make glucagon, then it is logical to think that the glucagon gene would be found in heterochromatin, right? And the insulin gene, because the beta cells express insulin, right? The insulin gene must be found in euchromatin, okay? Then whenever insulin is needed, right, after you have a heavy meal and all, then the insulin gene would be transcriptionally upregulated, right, by the binding of the activator to the enhancer, and of course the transcription initiation complex will be formed at the promoter, and so on. All right. So you can see how the control by the chromatin coiling, right, the previous lecture or previous video rather, that is more permanent, right? So because the glucagon gene is found in heterochromatin, the beta cells will never express glucagon, and the insulin gene will be found in the euchromatin, and the insulin gene is expressed whenever insulin is needed. Okay, so that's the difference between the two levels. The chromatin level, the control is more permanent and long-term, right? A cell that never express a particular gene, right? Then the gene should be in heterochromatin form. Then for the transcriptional control, it is for a gene that a cell will express. It is just controlling, like, exactly when it is expressed and how much. Okay, so this chromatin, a bit more on the chromatin coiling part, yeah. It is also observed when cells or stem cells differentiate. So if you think about stem cells, right, stem cells express telomerase, but when it is differentiated, right, it no longer expresses telomerase. Right, so you can imagine that the coiling of the chromatin will have changed during differentiation. Right, so at first, the telomerase gene should be found in euchromatin, but then during the process of differentiation, the coiling of the chromatin will have changed such that the part of the chromatin where the telomerase gene is found would have changed into heterochromatin, and therefore, once differentiated, the cell doesn't express telomerase anymore. Okay, so yeah, just wanted to differentiate the two levels of control a bit, even though both kind of control transcription. Okay, so anyway, let me continue, right? So we talked about the transcription factors, right? So as a protein, it has a particular structure, obviously. So structure always allows for function. So we will look a bit on the structure of a typical transcription factor and how that allows for function. All right. So on a transcription factor, right, it is a globular protein. It has specific shape and 3D conformation. Part of the 3D conformation, right, would be the DNA binding site or DNA binding domain. Domain for a protein just means a particular part of the protein. So it's a part that is responsible for DNA binding. Okay, so it must have a specific shape, right, to recognize a specific DNA sequence. So if it is an activated protein, then obviously that DNA binding domain will recognize an enhancer. If it is a repressor protein, it will recognize a silencer. Right? If it is a general transcription factor, then it recognizes the data box. Right? There would also be a activation domain that is responsible for interacting with other proteins in transcription. Okay, so if it is a activator protein, right, then it would interact with the transcription initiation complex to upregulate transcription. Repressor protein would interact with transcription initiation complex to downregulate transcription. Okay, so one part to bind to DNA and one part to bind to and interact with other proteins. Okay. Okay. So anyway, you have this picture in your notes, right? I just wanted to highlight a particular mistake in this picture, right? So this is supposed to be a specific transcription factor, right, that is interacting with the transcription initiation complex, as you can see here, interaction. But then, as you have learned before, right, if this is a specific transcription factor, then this particular site that it binds to would be either an enhancer or a silencer, right? Which should be far away from the gene and the promoter. So that should be the DNA looping, which is not shown here, right? So it shouldn't be so short a distance. It should be like very long, and then you have the DNA bending, DNA looping, right, to bring this either activator or repressor protein, right, close to the transcription initiation complex, right? So this DNA looping is not shown. Okay, just wanted to highlight that. Okay, let's continue with looking at a bit more on the repressor specifically, right? So as mentioned just now, right, besides the direct interaction between the repressor protein to the transcription initiation complex to downregulate transcription, which is part C that we see here, there are other mechanisms how the repressor can downregulate transcription, which is a bit different from the prokaryotic repressor. So even though both kind of cells have repressor protein, they really work differently, and they bind to different DNA sequences also. So anyway, for the repressor protein, other mechanisms include, right, preventing the binding of the activator to the enhancer, right? So you can see for A, right, this would be the enhancer sequence, this one would be the repressor sequence. They kind of overlap, or the silencer sequence, right? They kind of overlap a bit. Right? So when the repressor binds, then it blocks the binding of the activator. Okay? For B, right, it is to prevent the function of the activator. All right, so as you have looked at earlier, the activator should interact with the transcription initiation complex via the activation domain, right, to upregulate the rate of transcription. But in this case, what happened is the activation domain of the activator protein is blocked by the repressor. So there's no interaction with the transcription initiation complex to upregulate transcription. Okay. For part D and E, they seem similar. What happened is the repressor can recruit other proteins to form heterochromatin, and that would really, prevent transcription of the gene, right? Because there's no more access of the general transcription factors or RNA polymerase to the promoter. Okay. So whether it is recruitment of histone deacetylase that you have learned before, or some other kind of proteins called chromatin remodeling complex, is the result is the same. Heterochromatin is formed. Okay. All right. So a little bit more on the control elements. Well, so far we have been looking at just one enhancer, right, bound by one activator, or one silencer bound by one repressor. But in real life, right, a particular gene is controlled by more than one enhancers and silencers. Okay, so in my example here, right, we have this gene in this orange color, right, promoter is here, right, bound by the transcription initiation complex with the RNA polymerase here, right. It is controlled by 4 enhancers and 1 silencer. Okay, so this is not a must for all genes. Yeah, there are different combinations, where some can be 2 enhancers and 3 silencers, for example. So in this case, the example is 4 enhancers and 1 silencer. Okay, so as you can see, right, all the enhancers and silencer is bound by respective activators and repressor proteins. Okay, so now, right, transcription is upregulated by 4 activators and downregulated by 1 repressor. So this will give you a particular rate of transcription, right? So imagine that now we only have 3 activators binding and still have that 1 repressor binding. Then the rate of transcription would be slightly lower, right? Or, right, 1 less activator now, then even lower rate of transcription, versus now all 4 activators binding but no repressor binding. Then the rate of transcription will be very high. So you can see, right, when we have a combination of different control elements together controlling the transcription of the same gene, it allows for a very refined control, right? You can have, instead of only have transcription versus no transcription, we can have transcription, then transcription slightly faster, transcription even faster, gear 1, gear 2, gear 3, and so on. So there is what we call a fine control of the rate of transcription, a more precise control as well. Yeah. And at any one time in the cell, right, the presence of different activators and repressors would be different, because remember that activators and repressors are themselves proteins which are coded for by genes. So the activator genes and repressor genes can also be controlled, right? So at any one time, the type of and number of activators and repressors present in the nucleus would be different, and therefore that gives rise to different levels or different rates of transcription. So this is how the cell is able to control not only whether to have transcription or not, but also how much transcription at any one time, but depending on the combinations of transcription factors binding to the combination of control elements that control one particular gene. Okay, so it's a lot more complex and refined compared to the prokaryotes. So one example is this, right? In different cells, here liver cell versus the lens cell in the eye, right? You have a different set of transcription factors. So in the liver cell, the transcription factors are able to bind to the enhancers that control the albumin gene, but not the enhancers that control the crystalline gene. And therefore, the albumin gene is expressed in the liver cell, but not the crystalline gene. The reverse is happening in the lens cell, right? The set of transcription factors, activators specifically, the set of activators present binds to the enhancers of the crystalline gene, but not the albumin gene. So crystalline gene is expressed, but not the albumin gene. All right. So in these two cells, you can see both genes, right, albumin and crystalline, both genes are found in euchromatin. Right? If not, we won't be talking about binding of proteins, right? But because of the presence of different activators, then it controls the transcription of the two genes differently. Okay? So this is how a combination of control elements can upregulate or downregulate to different levels the transcription of different genes, right, in different cells at different times. Okay? Right, so yep, that would be the transcriptional control, right, of gene expression in eukaryotes.